Imagine yourself drifting, weightless in the velvet embrace of interstellar space. Around you, nebulae paint the darkness with strokes of cosmic dust and glowing gas, ancient nurseries where stars are born, and perhaps where the seeds of life are sown. Below, a nascent planet, a swirling sphere of cooling magma and primordial oceans hangs like a jewel. There's a profound silence here, yet it feels pregnant with possibility, as if the universe itself is holding its breath, anticipating the first tentative stirrings of something new, something alive. It is from this sense of immense scale and profound mystery that our journey tonight begins, a gentle exploration into the very origins of life and our search for cosmic kin. So, before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy they drift off. Now dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey together. We begin by softly questioning the very essence of what it means to be alive. What is this enigmatic phenomenon we call life? It seems at first glance an almost trivial question. You look at a silent, unmoving stone, and then at a bird taking flight, and the distinction feels utterly self-evident. The stone merely is, it endures, shaped by external forces, but it does not strive, it does not replicate, it does not grow in the same intrinsic way. The bird, however, breathes, eats, moves with purpose, seeks a mate, and raises its young, participating in an unbroken chain of existence stretching back countless generations. Yet as you delve deeper into this question, the seemingly sharp line between the living and the non-living begins to waver, to blur into a fascinating spectrum of complexity. Scientists and philosophers have long sought a precise, all-encompassing definition, but life, in its boundless ingenuity, often seems to delight in eluding our neat categorizations. We can, however, identify a collection of characteristics that most living things on Earth share.
One such characteristic is metabolism. This is the sum of all the chemical processes that occur within an organism to maintain life. It involves taking in energy and matter from the environment, sunlight for a plant, food for an animal, and converting it through intricate chemical reactions to build cellular components, to repair damage, and to provide the energy needed for all other life activities. Think of it as the organism's internal engine constantly running, processing fuel, and generating power and new power.
Then there is reproduction, the ability to create new individuals, passing on genetic information to offspring. This ensures the continuation of the lineage, the propagation of life through time. Whether it's a bacterium dividing into two, a flower producing seeds, or an animal giving birth, this fundamental drive to replicate is a hallmark of the living world. Without it, any spark of life would quickly extinguish. Growth and development are also key. Living things typically increase in size and complexity over their lifespan, following a pattern encoded in their genetic material. A tiny seed sprouts and grows into a towering tree. A single fertilized egg develops into a complex multicellular organism. This isn't just a passive accumulation of material, like a snowball rolling downhill. It's an organized, internally directed process of construction and transformation. Living entities also exhibit response to stimuli. They can sense changes in their external or internal environment and react accordingly. A plant might turn its leaves towards sunlight, a microbe might move away from a harmful chemical, an animal might flee from a predator. This responsiveness allows organisms to adapt to changing conditions, to seek out resources, and to avoid dangers, enhancing their chances of survival. Perhaps one of the most profound characteristics is evolution by natural selection. Populations of living organisms change over generations. Individuals with traits that are better suited to their environment are more likely to survive and reproduce, passing those advantageous traits to their offspring. Over vast stretches of time, this process can lead to the emergence of new species and the incredible diversity of life we see on Earth. This capacity for adaptive change is what allows life to persist and diversify in an ever-changing world. Another common feature is homeostasis, the ability to maintain a relatively stable internal environment, despite fluctuations in the external environment. For example, Your body works to keep its temperature, pH, and water balance within a narrow range, essential for the proper functioning of your cells. This internal regulation is crucial for survival, and underpinning many of these processes is organization. Living things are highly organized, from the molecular level, think of the intricate structure of DNA or proteins, to the cellular level, the complex organelles within a cell, and, in many cases, to the level of tissues, organs, and entire organisms. This intricate order is not accidental. It is essential for carrying out the complex functions of life. Now, while these characteristics provide a useful framework, you begin to see the blurriness when you encounter certain entities. Consider a virus. It contains genetic material, DNA or RNA, and can evolve. However, it lacks its own metabolism and cannot reproduce independently. It must infect a host cell and hijack its machinery to make copies of itself. Is a virus truly alive, or is it an incredibly sophisticated, non-living chemical package that merely mimics some aspects of life? Scientists still debate this, and the answer often depends on which definition of life you favor. What about a crystal? It can grow, adding new layers in a highly ordered pattern, and it certainly has organization. But it doesn't metabolize, reproduce in the biological sense, or evolve through natural selection. Or think of fire. It consumes fuel, a form of metabolism. It grows, it can reproduce by spreading, and it responds to its environment, e.g. wind. Yet, we intuitively know fire is not alive. It lacks the complex information-based replication and hereditary mechanisms of biological systems. This challenge of definition becomes even more acute when you consider the very earliest stages of abiogenesis, the origin of life from non-living matter. The first entities that could be considered proto-life might have possessed only a few of these characteristics in a very rudimentary form. Perhaps they were simple self-replicating molecules, not yet enclosed in a cell, with no metabolism to speak of. At what precise point in that gradual transition from complex chemistry to simple biology did the threshold into life truly get crossed? Was it a sharp line or an extended fuzzy dawn? As an analogy, imagine a spectrum of colors, from deep violet to vibrant red. It's easy to distinguish pure violet from pure red, but as you move along the spectrum, where exactly does violet end and blue begin? Where does green transition into yellow? There are intermediate shades, blended hues, where categorization becomes subjective. The transition from non-life to life might be similar, a continuous process with many intermediate stages, rather than a simple on-off switch. Philosophers and scientists still grapple with whether there can be a single universal definition of life that applies not only to all life on Earth, but also to any potential extraterrestrial life we might one day discover, which could be based on entirely different chemistry or operate under principles we haven't yet conceived. This very enigma, this gentle uncertainty about what life is, makes its origin all the more compelling. If the boundary is not absolute, then perhaps the chasm between the inanimate and the animate is not as impossibly wide as it sometimes seems. It suggests a continuum, a natural unfolding of complexity, where, given the right conditions and enough time, the universe can weave simple matter into the intricate, self-sustaining tapestry we call life. The exact processes involved, the specific chemical steps, and the environmental cradle that nurtured these first hesitant stirrings are what we will begin to explore as our journey continues. For now, let your mind rest on this foundational question, this beautiful ambiguity that lies at the very heart of existence. As you continue your weightless drift, your focus shifts more intently to that young, turbulent planet below. This is primordial Earth, some 4.5 billion years ago, a world almost alien to the lush blue marble we know today. To understand how life might have begun, you must first imagine the canvas upon which its earliest chemical sketches were drawn. The conditions were violent, yet paradoxically it was this very violence and instability that may have provided the necessary energy and raw materials for life's genesis. Picture a surface still intensely hot from its recent accretion, a molten or semi-molten landscape slowly cooling and solidifying into the first crustal rocks. The atmosphere, if one could even call it that in the earlier stages, would have been a roiling dynamic mixture, vastly different from the oxygen-rich air you breathe now. Instead of breathable oxygen, which is largely a byproduct of later life, photosynthesis, the early atmosphere was likely a reducing one, or perhaps neutral. This means it was probably rich in gases like water vapor, H2O, spewed forth from countless volcanoes, along with carbon dioxide, CO2, nitrogen, N2, and smaller amounts of gases like methane, CH4, ammonia, NH3, hydrogen sulfide, H2S, and hydrogen, H2. The exact composition is still a subject of intense scientific debate and research, as it has profound implications for the types of chemical reactions that could have readily occurred. Some models suggest a less reducing atmosphere, dominated more by CO2 and N2, similar to modern Mars or Venus. This detail matters immensely when scientists try to recreate early Earth conditions in the lab to test theories of abiogenesis. The energy sources bombarding this nascent world were immense and relentless. The young sun, though perhaps 30% fainter in overall luminosity than it is today, would have blazed with much higher levels of ultraviolet UV radiation. Earth had no protective ozone layer yet. Ozone, O3, is formed from oxygen, so this harsh UV light would have streamed down onto the surface and into the upper layers of any oceans, providing a powerful energy source capable of breaking chemical bonds and driving new reactions. Imagine a constant, invisible shower of high-energy photons, a cosmic catalyst. Then there was the lightning. With a turbulent atmosphere, likely choked with volcanic dust and water vapor, electrical storms would have been frequent and spectacular, far more intense and widespread than anything we experience today. Each colossal bolt of lightning would have discharged immense amounts of energy into the atmospheric gases, triggering chemical transformations, forging new molecules from simpler precursors. Picture the sky constantly illuminated by these primordial flashes, each one a miniature chemical factory. Volcanic activity itself was on a scale difficult to comprehend. The young Earth was still differentiating, its internal heat immense. Volcanoes, both on land, as land masses began to form, and beneath the forming oceans, would have continuously spewed out gases, contributing to the atmosphere and lava, building up the crust. These volcanic regions would have been hotspots of chemical activity, releasing minerals and chemical compounds from the planet's interior into the environment. Hydrothermal vents on the ocean floor, which we will explore in more detail later, are a modern analogue of this intense geothermal activity, releasing geothermally heated, mineral-rich water into the surrounding ocean. And let's not forget the cosmic bombardment. The early solar system was a far more chaotic place, a kind of cosmic shooting gallery. For hundreds of millions of years after its formation, Earth was subjected to the late heavy bombardment, a period of intense impacts by asteroids, comets, and other planetary debris. These impacts were not gentle. Large impactors could release unimaginable amounts of energy, enough to melt vast swathes of the crust, vaporize any oceans, and temporarily create a steam-filled atmosphere. While seemingly destructive, these impacts also delivered significant amounts of material to the young Earth, including water ice and, crucially, organic molecules formed in space, a topic we'll delve into later. Even the shockwaves from these impacts could have provided the energy and pressure needed to synthesize complex molecules from simpler ones. Imagine these cataclysmic events not just as destroyers, but also as cosmic delivery systems and chemical processes. The first oceans were beginning to form as the planet cooled enough for the water vapor outgassed from volcanoes to condense and fall as torrential planet-wide rain, filling the basins of the rugged, cratered landscape. These early oceans would have been warm, perhaps even hot in places due to geothermal activity, and their chemistry would have been different from today's saline seas. They would have been a repository for dissolved atmospheric gases and minerals leached from the primitive crust, creating a kind of planetary-scale chemical broth. So this primordial Earth was not a tranquil, serene garden. It was a dynamic, often violent and extreme environment. It was a world of fire and water, of intense radiation and electrical storms, of constant volcanic exhalations and devastating cosmic impacts. Yet it is precisely within this crucible of extremes that the conditions for life's origin are thought to have been forged. The very energies that could destroy could also create. The very materials spewed from volcanoes or delivered by comets could become the building blocks. Think of it like a blacksmith's forge. The blacksmith needs intense heat to make the metal malleable, powerful hammer blows to shape it, and specific raw materials to work with. Early Earth was this planetary-scale forge. The heat came from its formation, from radioactivity, from impacts. The hammer blows were the lightning, the UV radiation, the shock waves, and the raw materials were the simple gases and minerals present. It was a system far from equilibrium, constantly being churned and energized, providing countless opportunities for new chemical combinations to arise. Scientists still debate the precise timing and sequence of these early environmental conditions. For example, when did a stable crust form? When did the oceans reach a significant volume and a relatively stable composition? How intense and prolonged was the late heavy bombardment, and could it have repeatedly sterilized the planet, forcing life to restart multiple times or to take refuge in protected environments like deep underground or near hydrothermal vents? These are active areas of research, pieced together from geological evidence in the oldest surviving rocks on Earth, which are themselves rare and heavily altered, from studying meteorites, which are like time capsules from the early solar system, and from sophisticated computer models of planetary formation and evolution. The key takeaway is that the stage for life's origin was not set on a calm, peaceful world, but on a planet undergoing tremendous geological and atmospheric upheaval. This violence provided the necessary disequilibrium, the constant input of energy and fresh materials that could drive the complexification of chemistry. It was a canvas of raw, elemental power upon which the intricate chemistry of pre-life could begin its slow, tentative dance. This turbulent environment, far from being an impediment to life, may have been an essential prerequisite, providing the dynamic conditions needed to jolt matter out of its simpler states and onto the path towards biological organization. The very chaos held the seeds of order. As you picture this dramatic primordial world, you can almost feel the immense creative potential simmering just beneath its turbulent surface, waiting for the right ingredients and the right spark. Having painted a picture of primordial Earth's energetic and volatile canvas, let's now turn our attention to the specific paints themselves, the essential chemical elements and simple molecules that were available, the raw ingredients for life's intricate recipe. The universe, in its grand wisdom, provided a surprisingly limited yet incredibly versatile toolkit of elements from which to construct something as complex as a living cell. Of the 90-plus naturally occurring elements in the periodic table, only a handful make up the vast majority of living matter on Earth. The big four are carbon, C, hydrogen, H, oxygen, O, and nitrogen, N. Together, these four elements constitute about 96% of the human body's mass, and similar proportions are found in most other terrestrial organisms. Phosphorus, P, and sulfur, S, are also crucial, often included to make up the acronym CHNOPS, representing the 6 most important elements for life as we know it. Beyond these, a variety of other elements are needed in smaller or trace amounts, such as calcium, potassium, sodium, chlorine, magnesium, and iron, each playing vital roles in specific biological processes. But how did these elements find themselves on the early Earth, and in what forms were they available to participate in prebiotic chemistry? The story begins with the stars. Hydrogen and helium were formed in the Big Bang itself. All the other heavier elements, including the crucial carbon, nitrogen, and oxygen, were forged in the nuclear furnaces of earlier generations of stars. When these massive stars reached the end of their lives, they exploded as supernovae, scattering these newly synthesized elements across interstellar space. These elements then became incorporated into vast clouds of gas and dust, the nebulae from which new stars and planetary systems, including our own, would eventually form. So the very atoms that make up your body were once stardust, a rather poetic and profound connection to the cosmos. As the solar system formed, these elements condensed and accreted to form the planets. On early Earth, hydrogen was abundant, primarily locked up in water, H2O, but also present as molecular hydrogen, H2, in the early atmosphere, Though much of the light H2 gas would have escaped Earth's gravity over time, oxygen was overwhelmingly bound in water molecules and in silicate minerals within the planet's rocks. Free breathable oxygen, O2, was virtually absent until life itself began to produce it through photosynthesis much later. Nitrogen, a relatively inert gas, likely formed a significant component of the early atmosphere as molecular nitrogen, N2, much like it does today, about 78% of our current atmosphere. Carbon, the absolute backbone of life as we know it, is where things get particularly interesting. Carbon's remarkable ability to form 4 strong covalent bonds with other atoms, including itself, allows it to create an astonishing variety of complex molecules. Long chains, branch structures, and intricate rings. This molecular versatility is what makes carbon so well suited to be the structural basis of life. On early Earth, carbon would have been present in several forms. In the atmosphere, it existed as carbon dioxide, CO2, and methane, CH4. Dissolved in the early oceans, CO2 would have formed carbonic acid and carbonate ions. Carbon would also have been present in minerals within the crust and mantle. Furthermore, as we touched upon, meteorites and comets, remnants from the formation of the solar system, continuously delivered carbon-rich organic compounds to the early Earth. These extraterrestrial sources could have provided a significant starter kit of more complex carbon molecules. Phosphorus, essential for DNA, RNA in the phosphate backbone, and ATP, the cell's energy currency, would have been primarily derived from phosphate minerals in rocks. Its availability, particularly in a soluble form that could participate in chemical reactions, is considered by some scientists to be a potential limiting factor for the origin of life, as phosphate minerals are not always easily dissolved. Sulfur, a key component of some amino acids, like cysteine and methionine and certain vitamins, would have been available from volcanic gases like hydrogen sulfide, H2S, and sulfur dioxide, SO2, as well as from sulfide minerals. So you have these key elements present on the early Earth. But how did they combine to form the simple organic molecules that are the building blocks of life's larger structures? These include amino acids, the monomers that build proteins, nucleotides, the monomers for RNA and DNA, simple sugars, carbohydrates, and fatty acids, components of lipids which form cell membranes. This is where the energetic conditions we discussed in the previous section come into play. The UV radiation from the young sun, the electrical discharges from lightning, the heat from volcanoes, and the shock energy from impacts could all have provided the necessary activation energy to break the bonds of simpler inorganic molecules, like water, methane, ammonia, carbon dioxide, and allow their constituent atoms to recombine into these more complex organic monomers. The famous Miller-Urey experiment in 1952 was a landmark demonstration of this principle. Stanley Miller and Harold Urey passed electrical sparks, simulating lightning, through a mixture of gases, methane, ammonia, hydrogen, and water vapor, thought at the time to represent Earth's early atmosphere. After just a week, they found that a significant portion of the carbon had been converted into a variety of organic compounds, including several different amino acids. While the exact composition of the gas mixture used in the Miller-Urey experiment is now debated, some scientists favor a less reducing early atmosphere, richer in CO2 and N2, which yields fewer amino acids in such experiments. The experiment's fundamental importance remains. It showed that the building blocks of life could indeed arise from simple inorganic precursors through plausible natural processes. Subsequent experiments using different gas mixtures and energy sources have confirmed that a wide range of organic monomers can be synthesized under various simulated early Earth conditions. Think of it like a cosmic kitchen. The elements are your basic pantry staples: flour, sugar, eggs, etc. The simple inorganic molecules in the early atmosphere and oceans are like having those staples measured out into bowls. The energy sources, lightning, UV, volcanic heat, are like the oven, the mixer, and the stovetop. These appliances provide the energy to transform the raw ingredients into something more complex, like batter or dough. These are your organic monomers, like amino acids and nucleotides. Of course, simply forming these monomers is not enough. They then needed to become concentrated in specific locations and then link together to form polymers, long chains like proteins from amino acids, or nucleic acids like RNA from nucleotides. This polymerization is a crucial step, and how it occurred is another area of intense research. Perhaps it happened in evaporating shallow pools on the edges of volcanoes, where wetting and drying cycles could have concentrated the monomers and provided energy for them to link up. Mineral surfaces, particularly clays, might have played a vital role. Clay minerals have layered structures that can trap and concentrate organic molecules, and their surfaces can act as catalysts, facilitating the formation of polymer chains. Imagine these clay particles as microscopic assembly lines or templates. Scientists still debate the relative importance of different environments for the synthesis and accumulation of these prebiotic molecules. Was it primarily atmospheric synthesis driven by lightning and UV, as Miller-Urey suggested? Or did deep-sea hydrothermal vents, with their unique chemical gradients and mineral catalysts, play a more significant role, as some theories propose? Or was the delivery of organic molecules from space, via meteorites and comets, the dominant source of these crucial starting materials? It's quite likely that all of these processes contributed to varying degrees, creating a rich and diverse chemical inventory on the early Earth. The key concept here is spontaneous generation, but not in the old discredited sense of complex life like mice appearing from hay. Rather, it's the spontaneous formation of the chemical building blocks of life from simpler, non-living matter, driven by natural energy sources in a suitable environment. The early Earth provided both the diverse elemental ingredients and the energetic cooking processes to transform them into the molecular toolkit from which the first rudimentary forms of life could eventually self-assemble. Understanding this primordial chemical inventory is like understanding the available colors on an artist's palette before they begin to paint their masterpiece. The elements were there, the simple molecules began to form, and the stage was slowly being set for an even more extraordinary transformation. Having painted a picture of primordial Earth's energetic and volatile canvas, let's now turn our attention to the specific paints themselves, the essential chemical elements and simple molecules that were available, the raw ingredients for life's intricate recipe. The universe, in its grand wisdom, provided a surprisingly limited yet incredibly versatile toolkit of elements from which to construct something as complex as a living cell. Of the 90-plus naturally occurring elements in the periodic table, only a handful make up the vast majority of living matter on Earth. The big four are carbon, C, hydrogen, H, oxygen, O, and nitrogen, N. Together, these four elements constitute about 96% of the human body's mass, and similar proportions are found in most other terrestrial organisms. Phosphorus, P, and sulfur, S, are also crucial, often included to make up the acronym CNOPS, representing the 6 most important elements for life as we know it. Beyond these, a variety of other elements are needed in smaller or trace amounts, such as calcium, potassium, sodium, chlorine, magnesium, and iron, each playing vital roles in specific biological processes. But how did these elements find themselves on the early Earth, and in what forms were they available to participate in prebiotic chemistry? The story begins with the stars. Hydrogen and helium were formed in the Big Bang itself. All the other heavier elements, including the crucial carbon, nitrogen, and oxygen, were forged in the nuclear furnaces of earlier generations of stars. When these massive stars reached the end of their lives, they exploded as supernovae, scattering these newly synthesized elements across interstellar space. These elements then became incorporated into vast clouds of gas and dust, the nebulae from which new stars and planetary systems, including our own, would eventually form. So the very atoms that make up your body were once stardust, a rather poetic and profound connection to the cosmos. As the solar system formed, these elements condensed and accreted to form the planets. On early Earth, hydrogen was abundant, primarily locked up in water, H2O, but also present as molecular hydrogen, H2, in the early atmosphere, though much of the light H2 gas would have escaped Earth's gravity over time. Oxygen was overwhelmingly bound in water molecules and in silicate minerals within the planet's rocks. Free breathable oxygen, O2, was virtually absent until life itself began to produce it through photosynthesis much later. Nitrogen, a relatively inert gas, likely formed a significant component of the early atmosphere as molecular nitrogen, N2, much like it does today, about 78% of our current atmosphere. Carbon, the absolute backbone of life as we know it, is where things get particularly interesting. Carbon's remarkable ability to form 4 strong covalent bonds with other atoms, including itself, allows it to create an astonishing variety of complex molecules, long chains, branch structures, and intricate rings. This molecular versatility is what makes carbon so well suited to be the structural basis of life. On early Earth, carbon would have been present in several forms. In the atmosphere, it existed as carbon dioxide, CO2, and methane, CH4. Dissolved in the early oceans, CO2 would have formed carbonic acid and carbonate ions. Carbon would also have been present in minerals within the crust and mantle. Furthermore, as we touched upon, meteorites and comets, remnants from the formation of the solar system, continuously delivered carbon-rich organic compounds to the early Earth. These extraterrestrial sources could have provided a significant starter kit of more complex carbon molecules. Phosphorus, essential for DNA, RNA in the phosphate backbone, and ATP, the cell's energy currency, would have been primarily derived from phosphate minerals in rocks. Its availability, particularly in a soluble form that could participate in chemical reactions, is considered by some scientists to be a potential limiting factor for the origin of life, as phosphate minerals are not always easily dissolved. Sulfur, a key component of some amino acids, like cysteine and methionine and certain vitamins, would have been available from volcanic gases like hydrogen sulfide, H2S, and sulfur dioxide, SO2, as well as from sulfide minerals. So you have these key elements present on the early Earth. But how did they combine to form the simple organic molecules that are the building blocks of life's larger structures? These include amino acids, the monomers that build proteins, nucleotides, the monomers for RNA and DNA, simple sugars, carbohydrates, and fatty acids, components of lipids which form cell membranes. This is where the energetic conditions we discussed in the previous section come into play. The UV radiation from the young sun, the electrical discharges from lightning, the heat from volcanoes, and the shock energy from impacts could all have provided the necessary activation energy to break the bonds of simpler inorganic molecules, like water, methane, ammonia, carbon dioxide, and allow their constituent atoms to recombine into these more complex organic monomers. The famous Miller-Urey experiment in 1952 was a landmark demonstration of this principle. Stanley Miller and Harold Urey passed electrical sparks, simulating lightning, through a mixture of gases, methane, ammonia, hydrogen, and water vapor, thought at the time to represent Earth's early atmosphere. After just a week, they found that a significant portion of the carbon had been converted into a variety of organic compounds, including several different amino acids. While the exact composition of the gas mixture used in the Miller-Urey experiment is now debated, some scientists favor a less reducing early atmosphere, richer in CO2 and N2, which yields fewer amino acids in such experiments. The experiment's fundamental importance remains. It showed that the building blocks of life could indeed arise from simple inorganic precursors through plausible natural processes. Subsequent experiments using different gas mixtures and energy sources have confirmed that a wide range of organic monomers can be synthesized under various simulated early Earth conditions. Think of it like a cosmic kitchen. The elements are your basic pantry staples: flour, sugar, eggs, etc. The simple inorganic molecules in the early atmosphere and oceans are like having those staples measured out into bowls. The energy sources, lightning, UV, volcanic heat, are like the oven, the mixer, and the stovetop. These appliances provide the energy to transform the raw ingredients into something more complex, like batter or dough. These are your organic monomers, like amino acids and nucleotides. So, of course, simply forming these monomers is not enough. They then needed to become concentrated in specific locations and then linked together to form polymers, long chains like proteins from amino acids, or nucleic acids like RNA from nucleotides. This polymerization is a crucial step, and how it occurred is another area of intense research. Perhaps it happened in evaporating shallow pools on the edges of volcanoes, where wetting and drying cycles could have concentrated the monomers and provided energy for them to link up. Mineral surfaces, particularly clays, might have played a vital role. Clay minerals have layered structures that can trap and concentrate organic molecules, and their surfaces can act as catalysts, facilitating the formation of polymer chains. Imagine these clay particles as microscopic assembly lines or templates. Scientists still debate the relative importance of different environments for the synthesis and accumulation of these prebiotic molecules. Was it primarily atmospheric synthesis driven by lightning and UV, as Miller-Urey suggested? Or did deep-sea hydrothermal vents with their unique chemical gradients and mineral catalysts play a more significant role, as some theories propose? Or was the delivery of organic molecules from space via meteorites and comets the dominant source of these crucial starting materials? It's quite likely that all of these processes contributed to varying degrees, creating a rich and diverse chemical inventory on the early Earth. The key concept here is spontaneous generation, but not in the old discredited sense of complex life like mice appearing from hay. Rather, it's the spontaneous formation of the chemical building blocks of life from simpler, non-living matter, driven by natural energy sources in a suitable environment. The early Earth provided both the diverse elemental ingredients and the energetic cooking processes to transform them into the molecular toolkit from which the first rudimentary forms of life could eventually self-assemble. Understanding this primordial chemical inventory is like understanding the available colors on an artist's palette before they begin to paint their masterpiece. The elements were there, the simple molecules began to form, and the stage was slowly being set for an even more extraordinary transformation. While theories like the Miller-Urey experiment and the RNA world often evoke images of sun-drenched shallow pools or primordial atmospheric reactions, another compelling and dramatically different environment has been proposed as a potential cradle for life: deep-sea hydrothermal vents. Let's journey now from the Earth's surface, down through kilometers of dark, cold ocean water, to these remarkable oases of chemical activity on the abyssal plains. Here, far from the reach of sunlight, life might have found its first foothold, nurtured by the planet's own internal heat and chemistry. What exactly are these hydrothermal vents? They are essentially underwater hot springs, fissures in the ocean floor where seawater, having percolated deep into the oceanic crust, becomes superheated by underlying magma chambers and then erupts back into the ocean. As this hot water, which can reach temperatures exceeding 400 degrees Celsius or 750 degrees Fahrenheit in some black smoker vents, reacts with the surrounding volcanic rocks, it becomes enriched with dissolved minerals and chemicals such as hydrogen sulfide, H2S, methane, CH4, hydrogen, H2, iron, manganese, and other metals. When this hot mineral-laden fluid mixes with the near-freezing oxygen pore in the early ocean deep-sea water, many of these dissolved minerals precipitate out, forming towering chimney-like structures, often composed of sulfides and sulfates. These vents are dynamic environments, constantly spewing forth a rich chemical brew from Earth's interior. Could these dark, energy-rich environments have nurtured the first cells, providing a haven protected from the harsh conditions on early Earth's surface? The surface, as we've discussed, was bombarded by intense UV radiation and frequent large meteorite impacts that could have periodically sterilized shallow waters or even vaporized entire oceans. Deep-sea vents, however, would have been shielded from these surface hazards, offering a more stable and protected environment. They also provide a continuous source of chemical energy and a rich supply of reduced inorganic compounds, precisely the kinds of ingredients thought to be necessary for abiogenesis. One of the most striking discoveries supporting the potential of vents as cradles for life was finding vibrant, complex ecosystems thriving around modern-day hydrothermal vents, completely independent of sunlight. These communities, discovered in 1977, include giant tube worms, clams, mussels, crabs, shrimp, and a vast array of microorganisms. The base of this food web is not photosynthesis, which requires light, but chemosynthesis. Specialized bacteria and archaea, single-celled organisms that can look similar to bacteria but belong to a different domain of life, harness the chemical energy stored in compounds like hydrogen sulfide or methane gushing from the vents. They use this energy to convert inorganic carbon, like CO2 dissolved in the seawater, into organic matter, effectively eating the chemicals from the vents. This discovery revolutionized our understanding of where life could exist and how it could be powered, proving that sunlight is not an absolute prerequisite for complex ecosystems. If life can thrive there today, relying on geothermal chemistry, perhaps it first arose there as well. What types of minerals and organic compounds found near these vents could have acted as precursors to life or as catalysts for its formation? The vent structures themselves are fascinating in this regard. The porous mineral chimneys, with their labyrinthine internal structures and myriad tiny compartments, could have acted as natural chemical reactors. These compartments could have helped to concentrate precursor molecules, preventing them from dispersing into the vast ocean. The surfaces of minerals like iron sulfides, e.g. pyrite or fool's gold, clays, and other metal sulfides found in vent deposits can have catalytic properties, promoting the polymerization of amino acids into peptides or nucleotides into RNA-like chains. Think of these vent structures as naturally forming, flow-through chemical laboratories. The hot vent fluid provides a constant supply of reactants and energy, while the mineral surfaces provide catalytic sites and protected niches for reactions to occur. The steep thermal and chemical gradients between the hot, alkaline, in some types of vents, vent fluid, and the colder, more acidic, in the early Hadean Ocean, seawater could also have driven chemical reactions and even facilitated the formation of primitive metabolic cycles. Some theories, like Günther Wächtershäuser's iron-sulfur world hypothesis, propose that life began as a two-dimensional metabolic system on the surface of iron sulfide minerals, with the energy for fixing carbon derived from the formation of pyrite. There are different types of hydrothermal vents. The black smokers are very hot and acidic. Some scientists argue that these extreme temperatures might have been too destructive for the delicate molecules of early life. However, another type of vent, known as alkaline hydrothermal vents or white smokers, because their plumes are often rich in lighter-colored minerals like carbonates and silica, operate at lower temperatures, typically 40 to 90 BODs, and release fluids that are alkaline and rich in hydrogen gas. These conditions are considered by many to be particularly conducive to abiogenesis. The Lost City hydrothermal field in the mid-Atlantic is a prime example of such an alkaline system. The porous carbonate structures formed at these vents provide natural microcompartments and gradients that could have been ideal for the emergence of the first cells. The process of serpentinization, a geological reaction between seawater and mantle rocks that occurs in these settings, naturally produces hydrogen gas H2, methane CH4, and simple organic molecules, providing both energy and building blocks. How might life, if born in such isolated deep-sea oases, have eventually spread to colonize the rest of the planet? Initially, these vent-based life forms would have been localized. However, ocean currents, even in the deep sea, could have dispersed microbial cells or spores. Over geological timescales, as tectonic plates shifted and new vent fields formed and old ones died out, life could have hopped from one active vent system to another. Eventually, some of these early microbes might have evolved the ability to survive in the colder, surrounding deep-sea environment, or even to tolerate and eventually utilize sunlight, allowing them to ascend and colonize the vast photic zone of the upper oceans. This would have been a major evolutionary transition, opening up entirely new ecological niches. The exploration of hydrothermal vents on Earth continues to yield profound insights. We find extremophilic microorganisms thriving in these environments, pushing the known boundaries of life in terms of temperature, pressure, and chemical tolerance. These modern vent ecosystems serve as living laboratories, allowing us to study how life can adapt to and harness energy from purely geological sources. Moreover, the discovery of probable hydrothermal activity on icy moons like Saturn's Enceladus evidenced by plumes containing silica nanoparticles that form in hot water, makes these earthbound vents even more relevant as analogues for potential extraterrestrial habitats. If life can arise and thrive in such environments here, perhaps it can do so on other ocean worlds too. Scientists still actively debate the relative importance of deep-sea vents versus surface environments for life's origin. Some argue that the concentration of certain precursor molecules, like cyanide, a potential key ingredient in some prebiotic pathways, might be more favored in surface environments subject to UV radiation. Others point to the protection and continuous energy supply offered by vents. It's also possible that different stages of abiogenesis occurred in different locations. Perhaps some building blocks formed in the atmosphere or shallow pools and were then transported to vents where they became further organized, or vice versa. The puzzle of life's origin is complex, and it's likely that multiple environmental factors and locations played a role. However, the imagery of life emerging from these dark, warm, chemically rich fissures in the ocean floor is a powerful one. It paints a picture of life born not from gentle sunlight, but from the Earth's own fiery internal energy, a testament to the planet's inherent capacity to generate complexity from the bottom up. These vents remind us that even in the most seemingly inhospitable places, the drive towards life, or at least towards complex chemical organization, can be incredibly strong. As we ponder the various cauldrons on early Earth where life's ingredients might have brewed, be it sun-drenched shallow pools, the electrically charged atmosphere, or the chemically rich depths of hydrothermal vents, we must also lift our gaze to the cosmos itself. For it's entirely possible, and indeed increasingly considered likely, that some of the crucial building blocks for life, or perhaps even the very first primitive life forms, were not solely homegrown, but were delivered to our nascent planet from space. This brings us to the intriguing concept of panspermia and the more general idea of exogenous delivery of organic materials. The early solar system, as we've touched upon, was a far more dynamic and debris-strewn place than it is today. Comets, asteroids, and vast clouds of interplanetary dust particles, remnants from the solar system's formation, were abundant. Earth, in its youth, was constantly bombarded by this celestial rain. While very large impacts could be sterilizing, a steady influx of smaller meteorites, cometary fragments, and dust could have acted as a cosmic delivery service, enriching the early Earth with water and, significantly, a diverse array of organic molecules. Is there direct evidence for such extraterrestrial organic molecules? Absolutely. For decades, scientists have been analyzing meteorites that have fallen to Earth, particularly a class known as carbonaceous chondrites. These meteorites are like pristine time capsules from the early solar system, around 4.5 billion years old, and they have been found to contain a surprisingly rich inventory of organic compounds. It's crucial to understand that these organics are generally not considered to be remnants of life that existed within the meteorite itself, though that's a more speculative aspect of panspermia we'll touch on. Rather, they are complex molecules formed through non-biological, abiotic chemical processes occurring in interstellar molecular clouds in the protoplanetary disk from which the solar system formed, or on the asteroid parent bodies from which these meteorites originated. What kinds of molecules have been found? The list is impressive and highly relevant to the origin of life. Amino acids, the fundamental building blocks of proteins, are commonly found in these meteorites. Intriguingly, meteorites like the Murchison meteorite, which fell in Australia in 1969, contain a diverse suite of over 70 different amino acids, including many that are not used by life on Earth, as well as the 20 that are. Nucleobases, the critical information-carrying components of DNA and RNA, adenine, guanine, cytosine, thymine, and uracil, have also been identified in various meteorites. Sugars, including ribose, the sugar in RNA, and sugar-related molecules have been detected. Fatty acids, which can self-assemble into membrane-like vesicle structures, primitive cell boundaries, are also present. Polycyclic aromatic hydrocarbons, PAHs, complex carbon ring structures, are abundant as well. The presence of these vital prebiotic ingredients within meteorites provides strong evidence that the raw materials for life are not unique to Earth, but can form naturally throughout the cosmos and be delivered to planetary surfaces. This concept of exogenous delivery of building blocks is sometimes called molecular panspermia or soft panspermia. It doesn't necessarily mean that life itself arrived from space, but that the essential starter kit of organic molecules was significantly supplemented, or perhaps even primarily supplied, by extraterrestrial sources. This could have given a crucial kickstart to prebiotic chemistry on early Earth, providing a more diverse and abundant pool of monomers from which more complex structures could self-assemble. Now let's consider the more radical and classic form of panspermia, sometimes called lithopanspermia, life traveling within rocks, or interstellar panspermia. This is the hypothesis that viable living microorganisms, perhaps hardy bacterial spores or other extremophils, could be transported through space, embedded within meteorites or comets, and seed life on other habitable planets. The idea was popularized in modern times by astronomers Sir Fred Hoyle and Chandra Wickramasinghe, though its roots go back further. How might this happen? Imagine a large asteroid or comet impacting a planet that already harbors life. The force of the impact could eject vast quantities of rock and debris from the planet's surface into space, some of which might contain trapped microbes. If these rocks are large enough, their interiors could shield the microbes from the harsh conditions of space. The vacuum, the extreme temperature fluctuations, and the intense UV and cosmic radiation during a long interplanetary or even interstellar journey. If such a life-bearing rock eventually encountered another habitable planet and survived atmospheric entry, perhaps as a meteorite, it could release its microbial cargo, potentially seeding that new world with life. What are the challenges and supporting arguments for lithopanspermia? The challenges are significant. Microbes would need to survive the initial shock and acceleration of being ejected from their home planet. Then, they'd face the hazards of space for potentially thousands, millions, or even billions of years. Finally, they'd have to survive the heat and forces of atmospheric entry and impact on a new planet. However, we know of incredibly resilient microorganisms on Earth, often called extremophiles. Bacteria like Deinococcus radiodurans can withstand enormous doses of radiation and have remarkable DNA repair mechanisms. Bacterial spores can enter a dormant state, highly resistant to desiccation, vacuum, and temperature extremes for very long periods. Experiments exposing microbes to simulated space conditions or even actual space conditions on the outside of spacecraft have shown that some can survive, particularly if shielded by a thin layer of rock or dust. The distances involved are vast. Interplanetary transfer within a solar system, e.g. from Mars to Earth or vice versa, seems more plausible than interstellar transfer as the journey times are shorter. Indeed, we have found numerous meteorites on Earth that are definitively Martian in origin, e.g. ALH8 4001, which sparked debate about possible ancient Martian microfossils. This proves that rocks can travel between planets. Whether they can carry viable life is the key question. Interstellar panspermia is an even greater leap, requiring survival over light-year distances and much longer timescales. If panspermia were true, how would it change our understanding of life's origin? It wouldn't solve the fundamental problem of abiogenesis, how life arose from non-life in the first place. It would simply relocate the site of that origin to somewhere else in the universe. However, it would imply that life is far more common and interconnected than if it arose independently on every habitable world. Earth would be just one of many seeded planets. It could also mean that all life in a particular region of the galaxy might share a common ancient ancestor, spread like cosmic dandelion seeds on the stellar winds. Consider the analogy of islands in an ocean. Life could arise independently on each isolated island, spontaneous generation on each habitable planet. Or seeds and spores could be carried by wind and water currents from one island to another, colonizing new lands, panspermia. The truth might involve a combination of both. Perhaps the basic building blocks were widely delivered, molecular panspermia, making it easier for life to then arise independently on many worlds. Or perhaps in rarer cases, actual viable organisms made the journey. One intriguing piece of indirect evidence that sometimes comes up in discussions of panspermia is the handedness or chirality of biological molecules. Amino acids used by Earth life are almost exclusively left-handed, L-isomers, and sugars are right-handed, D-isomers. Interestingly, some carbonaceous meteorites have been found to contain a slight excess of certain L-amino acids. This has led to speculation that the initial bias towards L-amino acids on Earth might have been seeded or influenced by this extraterrestrial input, perhaps through exposure to circularly polarized light in interstellar space during the molecule's formation. If this preference was established in space and then delivered to Earth, it could have tipped the balance for terrestrial abiogenesis. Scientists still debate the overall contribution of extraterrestrial material to Earth's prebiotic inventory. Some argue it was a dominant source, while others believe Earth-based synthesis was more important. Regardless, the undeniable presence of complex organic molecules in meteorites and comets, and the ongoing rain of interplanetary dust, tells us that our planet is not an isolated chemical system. It has always been and continues to be connected to the wider cosmos. Whether that connection extended to the delivery of life itself remains one of the most captivating and open questions in our quest to understand our origins and our place among the stars. The cosmic kitchen, it seems, might have had ingredients delivered from very far away indeed. We've explored the chaotic yet generative conditions of early Earth, the essential chemical ingredients available, the intriguing possibility of an RNA-dominated biological era, the potential nurseries of life in deep-sea vents, and even the cosmic delivery of organic building blocks from space. Now we arrive at one of the most profound and still poorly understood transitions in the story of life. The monumental leap from a collection of complex organic molecules, amino acids, nucleotides, lipids, and sugars, however they were formed or delivered, to the first true self-replicating encapsulated cells. This is the threshold where chemistry truly becomes biology. Imagine that primordial environment, whether it's a shallow, sun-drenched pool, a porous rock crevice near a hydrothermal vent, or a microscopic water droplet within a clay matrix. Through the processes we've discussed, this environment has become enriched with the monomers of life. These monomers have, perhaps with the help of mineral catalysts or cycles of wetting and drying, begun to polymerize into longer chains, peptides, short proteins, strands of RNA, and perhaps simple lipids. But a loose collection of molecules, however complex, is not yet life. For that, at least two more critical developments were needed: compartmentalization, the formation of a boundary to separate an internal environment from the external world, and a reliable system for self-replication and heredity that could be passed down through generations. Let's first consider compartmentalization. Life as we know it requires a distinct internal environment where the complex chemical reactions of metabolism can occur in a controlled manner, concentrated and protected from the often harsh and unpredictable external surroundings. This is achieved by the cell membrane, a selectively permeable barrier that encloses the cell's contents. On modern Earth, cell membranes are primarily composed of phospholipids, molecules that have a water-loving, hydrophilic head and two water-fearing, hydrophobic tails. When placed in water, these molecules spontaneously self-assemble into a bilayer, with the hydrophobic tails tucked inwards away from the water, and the hydrophilic heads facing outwards towards the water on both the inside and outside of the cell. This creates a stable, flexible barrier. How might such membranes have formed prebiotically? Fatty acids and other lipid-like molecules, simpler than modern phospholipids, have been found in meteorites and can be synthesized under simulated early Earth conditions. These simpler amphiphilic molecules, having both water-loving and water-fearing parts, can also spontaneously form vesicles, small hollow spheres bounded by a membrane, when agitated in water. These protocells, or protobionts as they are sometimes called, would have been like tiny, naturally forming bubbles, encapsulating whatever molecules happened to be nearby in the primordial soup. They wouldn't have been true cells yet, lacking the complex machinery of modern life, but they would have represented a crucial first step towards individuality and a controlled internal environment. The formation of these vesicles could have had several advantages. Encapsulation would concentrate the reacting molecules, increasing the efficiency of chemical reactions. It would also protect these nascent biochemical systems from being diluted or destroyed by the external environment. Furthermore, if a vesicle happened to encapsulate a set of molecules that could, for example, draw in more building blocks from the outside or reinforce its own membrane, it would have a survival advantage and might grow and eventually divide, passing its successful contents to daughter vesicles. Now consider the challenge of self-replication and heredity. For life to persist and evolve, it needs a way to store information and to pass that information on to its offspring with reasonable fidelity, but also with the capacity for some variation, mutation, upon which natural selection can act. In the RNA world hypothesis, RNA molecules are proposed to have served this role, acting as both information carriers and catalysts. Imagine a protocell vesicle that happens to enclose a self-replicating RNA molecule, or a network of cooperating RNA molecules. If this RNA system could not only replicate itself, but also perhaps catalyze reactions that help to build or maintain the vesicle membrane or acquire resources from the environment, then this protocell would have a distinct advantage. It could grow, become unstable, and split into two daughter protocells, each carrying a copy or near copy of the successful RNA machinery. This would be the dawn of Darwinian evolution at the protocellular level. The exact mechanisms by which these processes occurred are still a subject of intense research and speculation. How did the first information-carrying polymers, like RNA, become reliably encapsulated within the first membranes? Did membranes form first and then later capture replicating molecules? Or did the replicating molecules somehow promote the formation of membranes around themselves? Some theories suggest that mineral surfaces, like clays, might have played a crucial role. Clays can absorb both lipids and nucleic acids, potentially bringing them into close proximity and facilitating their interaction. Perhaps protocells formed within the tiny pores of volcanic rocks or clay sediments, providing a scaffold and a protected microenvironment. Another major challenge is the development of even a rudimentary metabolism. Early protocells would have needed a way to harness energy from their environment to drive the synthesis of their components and maintain their organization against the constant tendency towards disorder, entropy. Perhaps they utilized simple chemical gradients across their membranes, or harnessed energy from reduced inorganic compounds like those found near hydrothermal vents, or even captured some energy from sunlight using simple pigment molecules. The first metabolic pathways would have been very simple, perhaps just a few coupled reactions, but they would have been essential for sustaining the protocell. The transition from a collection of independent replicating molecules to a system where these molecules are integrated within a bounded compartment, working together for the common good of the protocell, represents a major evolutionary transition in individuality. It's the shift from a society of genes to a society of cells. Think of it like this. You have individual musicians, the replicating molecules like RNA and catalytic molecules like ribozymes or simple peptides. Each musician can play their own tune, but when they come together inside a concert hall, the protocell vesicle, and begin to play in a coordinated way, perhaps following a simple shared rhythm, a primitive metabolic pathway, they create something much more complex and powerful, an orchestra, a cohesive entity. This orchestra can then, metaphorically, attract a larger audience, acquire resources, and perhaps even inspire the formation of new, similar orchestras. Scientists are exploring various scenarios in the laboratory. They can create lipid vesicles under plausible prebiotic conditions and study how they interact with RNA or proteins. They are trying to engineer synthetic, minimal cells to understand the fundamental requirements for a self-sustaining, self-replicating chemical system. Experiments have shown that RNA can be encapsulated within fatty acid vesicles, and that these vesicles can grow and divide under certain conditions, even without complex protein machinery. For instance, the addition of more fatty acids to the surrounding solution can cause vesicles to grow and elongate, and gentle physical forces, like shear from flowing water, can then cause them to split, distributing their internal contents. If those internal contents included replicating RNAs, then you have a system capable of rudimentary reproduction and heredity. The exact point at which a protocell could be considered truly alive is, like the definition of life itself, somewhat fuzzy. However, the emergence of a system that possesses a stable, selectively permeable boundary, a membrane, a genetic system capable of storing information and being replicated, e.g. RNA, a catalytic system to carry out essential reactions, e.g. ribozymes, a rudimentary way to harness energy, a simple metabolism, would represent a quantum leap towards life as we know it. This journey from a soup of molecules to the first organized, replicating protocells was likely not a single event, but a long, arduous process of chemical evolution, with countless false starts and dead ends. It was a period of intense molecular experimentation, driven by the laws of physics and chemistry, and filtered by the nascent forces of natural selection. The exact details remain one of the deepest and most challenging puzzles in science, but the overarching picture is one of increasing complexity and organization, a slow but persistent climb from the inanimate towards the animate, driven by the inherent properties of matter and energy in a dynamic early Earth environment. It's the story of how the universe, through countless tiny steps, began to build its first living machines. Having traced the remarkable journey from simple chemistry to the emergence of the first protocells on Earth, our voyage now expands outwards, seeking potential havens for life beyond our own planet. If life could arise here, given the right conditions, could it also arise elsewhere? This fundamental question drives the search for cosmic kin, but before we can search, we need an idea of what we're looking for. This brings us to the crucial concept of planetary habitability, and most prominently, the idea of the habitable zone around stars. What exactly defines a habitable world? In its broadest sense, a habitable environment is one that can sustain life. However, since Earth is the only place we definitively know life exists, our current search strategies are heavily biased towards finding conditions similar to those that support life as we know it, which is carbon-based and water-dependent. Therefore, when astronomers talk about habitable exoplanets, planets orbiting stars other than our Sun, they are usually referring to worlds that could potentially support liquid water on their surfaces. This focus on liquid water is paramount. Water is an extraordinary molecule with several properties that make it an ideal solvent and medium for life. It's a polar molecule, meaning it can dissolve a wide range of other substances, facilitating the complex chemical reactions necessary for biology. It's liquid over a relatively broad temperature range, particularly at pressures found on terrestrial planets. Its high heat capacity helps to moderate temperatures, preventing extreme fluctuations. Water also plays a direct role in many biochemical reactions. While we can speculate about life based in other solvents, a topic we might touch upon later, water remains our primary guide. This leads us to the concept of the stellar habitable zone, HZ, often more picturesquely called the Goldilocks zone, coined by astronomer Su-Shu Huang in 1959 and later popularized by others. The HZ is defined as the region around a star, within which an Earth-like planet with a suitable atmosphere could maintain liquid water on its surface. If a planet orbits too close to its star, the intense stellar radiation would cause surface water to boil away, perhaps leading to a runaway greenhouse effect like that seen on Venus, rendering the surface scorching hot and dry. If a planet orbits too far from its star, the surface temperatures would be so cold that water would freeze solid into ice, making it unavailable for biological processes. The habitable zone is that just right region in between, where surface temperatures could allow for lakes, rivers, and oceans of liquid water. The location and width of a star's habitable zone are not fixed. They depend critically on the type of star. Hotter, more luminous stars, like massive blue or white O, B, and A-type stars, emit far more energy, so their habitable zones will be much farther out and significantly wider. Cooler, dimmer stars, like M-type red dwarfs, which are the most common type of star in our galaxy, emit much less energy, so their habitable zones will be very close in to the star and much narrower. Our Sun, a G-type star, has a habitable zone that is often considered to encompass Earth comfortably, with Venus on its inner edge, though Venus is too hot due to its atmosphere, and Mars on its outer edge, generally too cold today for persistent surface liquid water, though it may have been within the HZ in its past. Think of it like sitting around a campfire. A roaring bonfire, a hot luminous star, requires you to sit much farther away to feel a comfortable warmth. A small, flickering campfire, a cool, dim star, requires you to huddle in very close. The comfortable warmth region is analogous to the habitable zone. However, simply being within the calculated habitable zone is not a guarantee of habitability. Several other factors play crucial roles. Planetary atmosphere. The presence and composition of an atmosphere are critical. A sufficiently dense atmosphere with greenhouse gases, like carbon dioxide, water vapor, and methane, is needed to trap some of the star's heat and maintain surface temperatures suitable for liquid water. Too little atmosphere, and the planet will be cold, and water will freeze or rapidly sublimate. Too much of certain greenhouse gases, and you can get a runaway effect like Venus. The atmosphere also provides pressure, which is necessary to keep water liquid at certain temperatures. Planetary size and mass. A planet needs to be large enough to have sufficient gravity to hold on to a substantial atmosphere over geological timescales. It also needs to be massive enough to retain internal heat from its formation and from radioactive decay, which can drive geological activity like volcanism and plate tectonics. Plate tectonics, as seen on Earth, is thought to play a vital role in regulating climate over long periods through the carbon-silicate cycle, which helps to stabilize atmospheric CO2 levels. If a planet is too small, like Mars or Mercury, it may lose its atmosphere and internal heat too quickly. If it's too large, it might become a gas giant or an ice giant, like Jupiter or Neptune, with no solid surface or with conditions entirely unsuitable for liquid water on a surface. Stellar activity. The host star's behavior matters. Young red dwarf stars, for instance, are known to be very active, frequently unleashing powerful stellar flares. Planets orbiting within the close-in habitable zones of these stars could be repeatedly bathed in intense X-ray and charged particle radiation, potentially stripping away their atmospheres or making their surfaces inhospitable unless they have strong protective magnetic fields. Planetary composition and structure. The presence of a rocky surface and the availability of key elements are important. A global magnetic field generated by a molten convecting core, like Earth's, is also considered highly beneficial as it shields the planet's atmosphere and surface from harmful solar wind and cosmic rays. Orbital stability and eccentricity. A planet's orbit needs to be relatively stable and not too eccentric. Elliptical. A highly eccentric orbit could take a planet through extreme temperature swings, moving it in and out of the habitable zone, making it difficult for stable liquid water to persist. It's also important to remember that the habitable zone is not static. It evolves as the star itself evolves. As a star like our Sun ages, it gradually becomes more luminous. This means its habitable zone slowly migrates outwards. Earth, currently well within the Sun's HZ, will eventually find itself on the hot inner edge in 1 billion years or so. Conversely, Mars might become warmer. For very long-lived red dwarf stars, their habitable zones remain relatively stable for trillions of years, potentially offering vast timescales for life to evolve, if the other challenges can be overcome. The concept of the habitable zone is a powerful tool for guiding our search for exoplanets that might harbor life, but it's crucial to recognize its limitations. It's primarily defined for surface liquid water and life as we know it. Life could potentially exist outside this zone, for instance in subsurface oceans on icy moons, like Europa or Enceladus in our own solar system, kept liquid by tidal heating or radioactive decay far from direct stellar radiation. Or, as we've speculated, life could be based on different chemistries or solvents, thriving in conditions we'd consider extreme. Scientists are continuously refining the definition and boundaries of the habitable zone, incorporating more complex atmospheric models and considering factors like cloud feedback, planetary rotation rate, and ocean circulation. The conservative habitable zone typically refers to a narrower region where liquid water could exist under a wider range of plausible atmospheric conditions, while the optimistic habitable zone extends these boundaries. The search for planets within these zones is a primary goal of exoplanet hunting missions, like NASA's Kepler Space Telescope, now retired, and the Transiting Exoplanet Survey Satellite, TESS, as well as upcoming observatories. Identifying these Goldilocks planets is the first step. The next, much harder step is to characterize their atmospheres and search for biosignatures, signs of life, a topic we will explore soon. For now, the habitable zone provides a crucial roadmap, helping us to prioritize targets in our vast cosmic neighborhood, focusing our attention on those worlds where the conditions for life's familiar song might just be right. Having defined the habitable zone as a prime target in our search for cosmic kin, the next immense challenge is actually finding these distant planets, or exoplanets, that orbit stars far beyond our Sun. Detecting a relatively small dim planet next to its incredibly bright host star from light years away is an astonishing feat of astronomical ingenuity. It's often likened to trying to spot a firefly hovering next to a distant searchlight, or hearing the whisper of a single instrument in a deafening cosmic orchestra. Astronomers have developed several clever indirect methods to achieve this, primarily by observing the subtle effects a planet has on its host star. One of the most successful and prolific techniques is the transit method. Imagine you are staring at a distant star, continuously and precisely measuring its brightness over time. If a planet happens to orbit that star, and if its orbital plane is aligned just right with our line of sight from Earth, an edge on orientation, then once per orbit, the planet will pass directly in front of its star from our perspective. This event is called a transit. When the planet transits, it blocks a tiny fraction of the starlight, causing a slight temporary dip in the star's observed brightness. The amount of dimming is proportional to the ratio of the planet's cross-sectional area to the star's area. So a larger planet will cause a deeper dip, allowing astronomers to estimate the planet's radius relative to its star. The duration of this transit dip gives clues about the planet's orbital speed and path. And if these dimming events repeat at regular intervals, that interval reveals the planet's orbital period, its year. From the orbital period and the star's mass, which can be estimated from its spectral type, Kepler's laws of planetary motion can be used to calculate the semi-major axis of the planet's orbit, or its average distance from the star. This is crucial for determining if the planet lies within the star's habitable zone. NASA's Kepler Space Telescope, launched in 2009, famously used the transit method to discover thousands of exoplanets by photometrically monitoring the brightness of over 150,000 stars simultaneously in a fixed patch of the sky. The Transiting Exoplanet Survey Satellite, TESS, launched in 2018, is now conducting an all-sky survey, looking for transiting planets around nearby, bright stars which are ideal targets for follow-up atmospheric characterization. The transit method requires precise continuous monitoring and sophisticated data analysis to detect these minute dips and rule out other causes of stellar variability, like star spots or instrumental effects. Another powerful technique, which led to the first confirmed discovery of an exoplanet around a Sun-like star, 51 Pegasi b in 1995, is the radial velocity method, also known as the Doppler spectroscopy or wobble method. As a planet orbits its star, its gravity doesn't just pull on the planet. The planet also exerts a gravitational tug on the star. Both the star and the planet actually orbit around their common center of mass, called the barycenter. Because the star is typically much more massive than its planets, this barycenter is usually very close to the star's center, but not exactly at it. This means the star itself executes a small orbit, or wobbles in response to its unseen planetary companions. We can detect this stellar wobble by observing the star's light using high-resolution spectroscopy. As the star moves slightly towards us in its tiny orbit, its light waves get compressed, shifting its spectral lines towards the blue end of the spectrum, a blue shift. As it moves slightly away from us, its light waves get stretched, shifting its spectral lines towards the red end of the spectrum, a redshift. This is the Doppler effect. By precisely measuring these tiny periodic shifts in the star's spectral lines over time, astronomers can infer the presence of an orbiting planet. The amplitude of this radial velocity variation reveals the planet's minimum mass. We can only determine m sin I, where m is the mass and I is the inclination of the orbit, since we usually don't know the inclination unless the planet also transits. The period of the wobble gives the planet's orbital period. The radial velocity method is more sensitive to massive planets orbiting close to their stars, as these produce a larger, more easily detectable wobble. While transits and radial velocity are the workhorses of exoplanet detection, other methods also contribute valuable discoveries. Direct imaging. This is the holy grail to actually taking a picture of an exoplanet. However, it's incredibly challenging due to the overwhelming glare of the host star, which can be millions to billions of times brighter than the light reflected by its planet. It's like trying to see a firefly next to a searchlight. Astronomers use sophisticated techniques like adaptive optics to correct for atmospheric blurring from Earth-based telescopes, and coronagraphs to block out the starlight, creating an artificial eclipse, or star shades. Direct imaging is currently most successful for detecting large Jupiter-sized planets that are relatively far from their host stars and are young enough to still be glowing brightly with the heat of their formation, making them more visible in infrared light. Capturing a direct image of a small rocky Earth-like planet in the habitable zone of a Sun-like star is a major goal for future, extremely large telescopes. Gravitational microlensing. This method relies on a phenomenon predicted by Einstein's theory of general relativity. If a star with a planet passes almost directly in front of a more distant background star, from our viewpoint, the gravity of the foreground star and its planet can act like a lens, bending and magnifying the light from the background star. The planet can cause an additional brief brightening or distortion in the magnification pattern. Microlensing events are rare and require precise alignment, so they often involve monitoring millions of stars. However, this technique is unique in its ability to detect planets at very large distances from their stars, even rogue planets that have been ejected from their star systems and wander through space alone. It's also sensitive to low-mass planets. Astrometry. This is the precise measurement of a star's position on the sky. Just as the radial velocity method detects the star's wobble along our line of sight, astrometry aims to detect the star's tiny side-to-side wobble on the plane of the sky due to the gravitational pull of an orbiting planet. This is an extremely challenging technique, requiring incredibly precise measurements over many years, but missions like the European Space Agency's Gaia satellite are making significant progress. Each detection method has its own strengths, weaknesses, and biases. For example, the transit method requires a specific orbital alignment, so it only finds a fraction of existing planets. The radial velocity method is less sensitive to smaller, more distant planets. By combining data from multiple methods, astronomers can get a more complete picture of an exoplanet system. If a planet is observed to both transit and have its radial velocity measured, scientists can determine both its radius, from the transit, and its mass, from radial velocity, which allows them to calculate its density. A planet's density is a crucial clue to its bulk composition, whether it's likely to be a rocky terrestrial planet like Earth, a gas giant like Jupiter, or an ice giant like Neptune. The thousands of exoplanets discovered so far have revealed an astonishing diversity of planetary systems, many quite different from our own solar system. We've found hot Jupiters, gas giants orbiting incredibly close to their stars, super-Earths, planets larger than Earth but smaller than Neptune, which could be rocky or have thick atmospheres, mini-Neptunes, planets orbiting binary stars, and systems with multiple planets in compact configurations. This incredible variety constantly challenges and refines our theories of how planets form and evolve. Think of these detection techniques as different ways of listening for planets. The transit method is like noticing when someone briefly walks in front of a light. The radial velocity method is like hearing the subtle change in a singer's voice as they sway back and forth. Direct imaging is like finally catching a glimpse of the performer. Each method gives us a different piece of information, and together they help us build a census of the planets in our galactic neighborhood, bringing us ever closer to identifying those potentially habitable worlds where our cosmic kin might reside. The ingenuity behind these methods is a testament to human curiosity and our relentless drive to explore the unseen. Having explored how we find distant worlds, let's now bring our focus back a little closer to home, to a planet that has captivated human imagination for centuries as a potential abode for life, Mars. Our rusty red neighbor, the fourth planet from the Sun, presents a tantalizing case study in planetary evolution and the potential for life to arise and perhaps even persist in environments different from Earth's. As we sift through the evidence gathered by orbiters, landers, and rovers, a picture emerges of a Mars that was once very different from the cold, arid desert we see today. A Mars that may have been warmer, wetter, and potentially more hospitable to life. What compelling geological features on Mars whisper of this ancient, more clement past? Decades of exploration have revealed a landscape carved by processes that strongly suggest the past presence of abundant liquid water. Orbiters like NASA's Mars Reconnaissance Orbiter, MRO, have mapped vast networks of dried-up riverbeds, valleys, and deltas that bear a striking resemblance to fluvial features on Earth. We see channels that meander, branch, and merge, indicative of sustained water flow. Perseverance rover is currently exploring Jezero crater, which is believed to have once held a lake, complete with an ancient river delta where water deposited sediments. Curiosity rover in Gale crater has found extensive evidence of ancient lake beds and streams. Layered sedimentary rocks, similar to those formed in water on Earth, are common in many regions. Beyond the large-scale landforms, specific minerals detected on the Martian surface provide further ground truth. Instruments on orbiters and rovers have identified hydrated minerals, such as clays, phyllosilicates, and sulfates. These minerals typically form in the presence of water over extended periods. Clays, for instance, suggest chemical weathering of rocks by water, while sulfates might indicate a period when waters were evaporating, leaving behind these salty deposits. The distribution and types of these minerals help scientists reconstruct the history of water on Mars and identify regions that might have been particularly habitable. When might Mars have been this warmer, wetter world? The evidence primarily points to the Noachian period, an early era in Martian history, roughly between 4.1 and 3.7 billion years ago. During this time, Mars is thought to have had a thicker atmosphere, likely richer in greenhouse gases like carbon dioxide, which could have trapped more of the sun's heat and maintained temperatures above freezing, allowing liquid water to be stable on the surface. This period on Mars significantly overlaps with the time when life is believed to have been emerging on early Earth, raising the tantalizing possibility that two neighboring planets in our solar system might have simultaneously possessed conditions suitable for abiogenesis. Some evidence suggests that intermittent water activity, perhaps in the form of episodic floods or seasonal melting, might have continued into the later Hesperian period, roughly 3.7 to 3.0 billion years ago. But by the current Amazonian period, Mars had largely transitioned into the cold, dry, and irradiated planet we observe today. This naturally leads to the profound question: if early Mars had liquid water, a primordial atmosphere, available energy sources, sunlight, geothermal heat, and the necessary chemical building blocks, potentially delivered by meteorites and comets, just as on Earth, or formed through Martian geological processes, could life have arisen there? It's certainly a plausible scenario. If the conditions were right, then the same chemical processes that led to life on Earth might have also occurred on Mars. This hypothetical Martian life would likely have been microbial, perhaps similar to Earth's earliest bacteria or archaea, adapted to the specific conditions of its environment. If life did emerge on ancient Mars, could it have survived the planet's dramatic climate change? As Mars lost its global magnetic field, which it appears to have had very early in its history, but which then dissipated, its atmosphere was gradually stripped away by the solar wind. Without a substantial atmosphere, the greenhouse effect weakened, the planet cooled, and liquid water on the surface became unstable, either freezing or sublimating into the thin air. If life had gained a foothold, it would have faced immense environmental pressure. However, life on Earth has shown incredible resilience and adaptability. Martian life might have retreated underground, seeking refuge in subsurface aquifers where liquid water could persist, kept liquid by geothermal heat or the presence of salts, forming brines that have a lower freezing point. We find thriving microbial ecosystems deep within Earth's crust and in subglacial lakes, so the idea of Martian life persisting in such hidden niches is not far-fetched. Rovers are looking for signs of past or present methane in the Martian atmosphere, as methane can be a byproduct of microbial metabolism, though it can also be produced by geological processes, so detecting localized or seasonal methane plumes could be an indirect hint of subsurface activity. What specific signs or biosignatures are our robotic explorers searching for that could indicate past or present Martian life? Organic molecules, particularly complex organic molecules or specific patterns of simpler ones that are unlikely to form through purely non-biological processes. Rovers like Curiosity and Perseverance have found simple organic molecules, like thiophenes, benzene, toluene, and small carbon chains, in ancient Martian mudstones. However, these can also have non-biological origins, e.g., from meteoritic infall or geological processes like serpentinization. The challenge is to find more complex, unambiguously biological organics, or patterns that point to a biological source. Microfossils and stromatolites, preserved physical remnants of microbial cells or their structures. This would be incredibly exciting, but also very difficult to confirm, as non-biological mineral formations can sometimes mimic simple biological shapes. Stromatolites are layered rock structures formed by the growth of microbial mats in shallow water. Finding their Martian equivalents would be strong evidence. Isotopic ratios. Living organisms on Earth preferentially use certain isotopes of elements like carbon, e.g., favoring carbon-12 over carbon-13. Unusual isotopic ratios in Martian carbon-containing compounds or minerals could indicate past biological processing. Mineralogical evidence. Certain minerals or mineral assemblages can be formed or altered by biological activity. Evidence of past habitable environments. Identifying locations that clearly had long-lived bodies of water, stable temperatures, and the necessary chemical ingredients makes those locations prime targets for searching for other biosignatures. Martian meteorites, rocks blasted off Mars by ancient impacts that eventually landed on Earth, have provided another avenue for study. The most famous is ALH 4001, found in Antarctica. In 1996, a team of NASA scientists announced that it contained several features: carbonate globules, specific organic molecules called PAHs, tiny magnetite crystals of a particular shape, and microscopic ovoid structures that they interpreted as potential evidence of ancient Martian microbial life. This claim sparked intense scientific debate. While the consensus now is that most, if not all, of these features in ALH 84001 can be explained by non-biological processes, the meteorite undeniably spurred interest and research in astrobiology and the search for life on Mars. It highlighted the kinds of features to look for and the extreme caution needed in interpreting them. The ongoing Perseverance rover mission is particularly significant because it is collecting and caching samples of Martian rock and soil that are planned to be returned to Earth by a future joint NASA-ESA, European Space Agency, Mars Sample Return campaign in the 2030s. Analyzing these pristine samples in sophisticated laboratories on Earth with instruments far more capable than anything we can send to Mars will offer our best chance yet to definitively answer the question of whether life ever existed on our planetary neighbor. Imagine Mars not just as a dusty red ball, but as a planet with a rich history, a world that may have once harbored flowing rivers, deep lakes, and perhaps, just perhaps, the quiet hum of primitive life. Sifting through its ancient rocks and dust is like planetary archaeology, searching for the faint echoes of a lost biosphere. Finding even the fossilized remains of simple microbes on Mars would be a discovery of profound significance, suggesting that life is not a unique fluke of Earth, but can arise independently on other worlds when conditions are right. It would mean that the answer to "Are we alone?" might be found right next door, in cosmic terms. Beyond the tantalizing, albeit now arid landscapes of Mars, our solar system beckons us towards even more exotic and potentially life-harboring environments, the icy moons orbiting the gas giant planets, Jupiter and Saturn. These worlds, far from the sun's warmth and traditional habitable zone, were long thought to be frozen, inert bodies. However, discoveries over the past few decades have radically transformed this view. revealing that several of these moons likely conceal vast subsurface oceans of liquid water beneath their icy shells. This opens up the astonishing possibility that life could exist in these dark, hidden seas, powered not by sunlight, but by chemical energy from the moon's interiors. Let's begin with Europa, one of Jupiter's 4 large Galilean moons. Europa's surface is a strikingly smooth, bright expanse of water ice, crisscrossed by a complex network of reddish-brown linear features called lineae, and areas of chaotic terrain where icy plates appear to have broken apart and shifted. This relatively young and geologically active surface strongly hints at a dynamic interior. Data from the Galileo spacecraft in the 1990s, including measurements of Europa's induced magnetic field, which suggests the presence of a global, electrically conductive layer, like a salty ocean, combined with theoretical modeling and Hubble Space Telescope observations of potential water vapor plumes, have built a compelling case for a global saltwater ocean beneath an icy crust estimated to be perhaps 10 to 30 kilometers, 6 to 19 miles, thick. This ocean could be up to 100 kilometers, 60 miles deep, potentially containing more than twice the volume of all of Earth's oceans combined. What keeps this ocean liquid so far from the sun, where surface temperatures on Europa are around 160 degrees, 260 degrees? The primary energy source is tidal heating. Europa is locked in an orbital resonance with 2 other large Jovian moons, Io and Ganymede. As Europa orbits Jupiter, the gravitational pulls from Jupiter and these other moons constantly flex and squeeze Europa's interior. This continuous tidal deformation generates immense friction and heat within the moon, much like repeatedly bending a paperclip makes it warm. This internal heat is believed to be sufficient to maintain the subsurface ocean in a liquid state, and could also drive geological activity on the ocean floor, such as volcanism or hydrothermal vents. Now let's journey to the Saturnian system and encounter Enceladus, a much smaller icy moon that has provided some of the most dramatic evidence for a subsurface ocean. The Cassini spacecraft, during its mission orbiting Saturn, made the astonishing discovery of enormous plumes of water ice particles, water vapor, and simple organic molecules erupting from tiger-stripe fissures near Enceladus's south pole. These plumes jet hundreds of kilometers out into space, indicating a pressurized subsurface source. Cassini flew directly through these plumes multiple times, tasting their composition. Analysis revealed that in addition to water and organics, like methane, the plumes contain salts, like sodium chloride, indicating a salty ocean, and, crucially, tiny silica nanoparticles. On Earth, such silica nanoparticles typically form when hot water, above 90 degrees or 194 degrees, interacts with rock, a strong indicator of active hydrothermal vents on Enceladus's ocean floor. The ocean itself is thought to be global, sandwiched between the icy shell and a rocky core. And what about Titan, Saturn's largest moon? Titan is unique in our solar system for possessing a dense nitrogen-rich atmosphere, even thicker than Earth's, complete with clouds, rain, rivers, lakes and seas. However, on Titan's frigid surface, around 179 degrees C or 290 degrees F, the liquid is not water, but hydrocarbons like methane and ethane. While its surface is inhospitable to water-based life, models suggest that Titan, too, likely harbors a subsurface ocean of liquid water, possibly mixed with ammonia, which acts as an antifreeze, buried deep beneath its icy crust and hydrocarbon surface. The energy source here is also likely a combination of tidal heating from Saturn and radiogenic heat from the decay of radioactive elements in its rocky core. Could these hidden oceans, particularly those of Europa and Enceladus, harbor life? The conditions seem potentially very favorable, especially if hydrothermal vents are active on their seafloors. Remember our discussion of terrestrial hydrothermal vents. They support thriving ecosystems based on chemosynthesis, where microbes derive energy from chemicals released from the planet's interior. Similar vents on the floors of Europa's or Enceladus's oceans could provide the necessary chemical energy, releasing compounds like hydrogen, methane, and sulfides, and dissolved minerals to support an independent origin and sustenance of life, completely in the absence of sunlight. The organic molecules detected in Enceladus's plumes further bolster this possibility, suggesting that at least some of the necessary building blocks for life are present. If life exists in these dark, cold, high-pressure environments, what might it look like? It would almost certainly be microbial, perhaps analogous to the bacteria and archaea found in Earth's deep-sea vent ecosystems or within our planet's deep biosphere. These organisms would need to be extremophiles, adapted to survive without light, at high pressures, and potentially at low temperatures, though it would be warmer near active vents. Their metabolism would be chemosynthetic. We wouldn't expect complex multicellular organisms like fish or squid, given the likely energy limitations and the challenges of evolving complexity in such environments. But even discovering simple, single-celled life would be a revolutionary find. It would demonstrate that life can arise and persist in conditions vastly different from Earth's surface, and that the necessary conditions for life are not limited to planets orbiting within a star's traditional habitable zone. The exploration of these ocean worlds is a high priority for space agencies. NASA's Europa Clipper mission is scheduled to launch in October 2024 and will arrive at Jupiter in 2030. It will perform dozens of close flybys of Europa, using a suite of instruments including ice-penetrating radar to confirm the ocean's existence, measure its depth and salinity, characterize the ice shell's thickness, and search for and analyze any active plumes. Looking further ahead, there are ambitious concepts for lander missions that could one day attempt to drill or melt their way through Europa's thick, icy shell using a cryobot, and deploy a submersible robotic probe, a hydrobot, to directly explore the ocean and search for signs of life. Such a mission is technologically daunting and likely decades away, but it represents an ultimate goal. For Enceladus, future missions might aim to fly through its plumes at lower altitudes, and with more sensitive instruments, to better analyze the ocean's composition and search for more complex organic molecules or even intact microbial cells. A lander mission near the South Polar fissures could also collect fresh plume material for in situ analysis. Titan is also a target for future exploration, with missions like NASA's Dragonfly, a rotorcraft lander set to launch in the late 2020s, which will explore Titan's surface chemistry and assess its habitability, potentially even sampling materials that could give clues about its subsurface ocean. Imagine plunging beneath the fractured icy crust of Europa or Enceladus. You pass through kilometers of solid, ancient ice until finally you break through into a vast, dark ocean. The only light might come from the eerie glow of hydrothermal vents on the distant seafloor, spewing out their chemical-rich plumes. Could there be life here, huddled around these oases of warmth and energy, a completely separate genesis from life on Earth? The allure of these hidden oceans is immense. They represent some of the most promising and accessible, compared to exoplanets, locations in our solar system to search for extant extraterrestrial life. Their discovery has fundamentally expanded our definition of what constitutes a habitable environment, pushing the boundaries far beyond sunlit surfaces and into the cold, dark depths of these enigmatic icy worlds. Our cosmic voyage has taken us from the potential cradles of life on early Earth and our solar system neighbors to the vast expanse of interstellar space, where countless exoplanets orbit distant stars. We've discussed how we find these remote worlds and how we identify those that might lie within their star's habitable zone. But once we've found a promising candidate, perhaps a rocky planet of similar size to Earth, orbiting at the right distance from its star to potentially host liquid water, how do we search for signs of life across light years of empty space? We can't send probes there, at least not with any current or foreseeable technology. Instead, we must rely on analyzing the faint light that reaches us from these systems, searching for subtle clues in planetary atmospheres known as biosignatures. A biosignature, in the context of astrobiology, is any substance, feature, or phenomenon that provides scientific evidence of past or present life. When we study exoplanet atmospheres, we are primarily looking for atmospheric biosignatures, specific gases or combinations of gases that, in the quantities detected, are difficult to explain by purely non-biological, geological, or photochemical processes alone, and might therefore indicate the presence of a global biosphere. One of the most sought-after atmospheric biosignatures is oxygen, O2. On Earth, the vast majority of the free oxygen in our atmosphere, about 21%, is produced by photosynthetic organisms, plants, algae, and cyanobacteria, which convert carbon dioxide and water into organic matter and oxygen, using sunlight as an energy source. While small amounts of oxygen can be produced abiotically, e.g., by the photolysis of water by UV radiation, especially if a planet is losing its water to space, sustaining a dense, oxygen-rich atmosphere, like Earth's over geological timescales, is thought to require a powerful, continuous biological source. Therefore, detecting significant levels of oxygen in an exoplanet's atmosphere would be a very compelling, though not definitive, sign of life. Closely related to oxygen is ozone, O3. Ozone is formed in the upper atmosphere when UV radiation from the host star interacts with oxygen molecules. The presence of a substantial ozone layer, detectable by its strong absorption of UV light, would imply the presence of a significant amount of oxygen below it. Ozone itself is also a potential biosignature, and it has the advantage that its spectral signature can sometimes be easier to detect than that of oxygen, especially from ground-based telescopes. Another key gas is methane, CH4. On Earth, a large portion of atmospheric methane is produced by methanogenic archaea, microbes that live in anaerobic, oxygen-poor environments like wetlands, rice paddies, and the digestive tracts of animals. Methane can also be produced by geological processes such as serpentinization, the alteration of certain types of rock by water, which we discussed in the context of hydrothermal vents. However, oxygen and methane are chemically reactive with each other. In an oxygen-rich atmosphere like Earth's, methane should be relatively quickly oxidized and destroyed. The fact that Earth has both abundant oxygen and a detectable amount of methane simultaneously implies that both gases are being continuously replenished. Methane, in this context, is in a state of chemical disequilibrium with the oxygen. Detecting such a disequilibrium, for example, the co-occurrence of oxygen and methane in significant quantities in an exoplanet's atmosphere, would be a particularly strong biosignature, as it's hard to explain without invoking a constant biological production of one or both gases. Other potential gaseous biosignatures include nitrous oxide, N2O, which is produced by denitrifying bacteria on Earth, and various sulfur compounds like dimethyl sulfide, DMS, produced by marine phytoplankton. The challenge with many of these is that they are typically present in much lower concentrations than oxygen or methane, making them harder to detect across interstellar distances. How do astronomers actually detect these gases in the atmospheres of distant exoplanets? The primary technique is transmission spectroscopy, which we touched upon when discussing exoplanet detection. When a planet transits, passes in front of, its host star from our point of view, some of the starlight passes through the planet's atmosphere, if it has one, on its way to our telescopes. Different molecules in the atmosphere absorb starlight at specific, characteristic wavelengths, colors. By carefully analyzing the spectrum of the starlight during a transit and comparing it to the spectrum of the star when the planet is not transiting, astronomers can identify these absorption features and deduce the presence and relative abundance of various gases in the planet's atmosphere. This is an incredibly delicate measurement, as the atmospheric signal is tiny compared to the overwhelming light from the star. Another technique is emission spectroscopy. This involves analyzing the light emitted directly by the planet itself, or light reflected from its surface and atmosphere. This is often done when the planet is observed just before or just after it passes behind its star, a secondary eclipse, allowing astronomers to subtract the starlight and isolate the planet's own thermal emission or reflected light spectrum. Powerful new telescopes like the James Webb Space Telescope, JWST, are revolutionizing our ability to perform these atmospheric characterizations. JWST's large mirror and its sensitive infrared instruments are particularly well suited for studying the atmospheres of transiting exoplanets, especially those orbiting cooler M-dwarf stars, where the habitable zones are closer in and transits are more frequent and produce a relatively larger signal. JWST is capable of detecting water vapor, methane, carbon dioxide, carbon monoxide, and potentially even biosignatures like oxygen or ozone in the atmospheres of some of the most promising nearby exoplanets. However, interpreting potential biosignatures is fraught with challenges. We must be extremely cautious about false positives, situations where a non-biological process could mimic a biosignature. For example, as mentioned, oxygen can be produced abiotically under certain extreme conditions, such as on a planet with a very low-pressure atmosphere that is losing vast amounts of water vapor, which is then split by UV radiation into hydrogen, which escapes to space, and oxygen, which might accumulate. Similarly, methane can be produced geologically. Therefore, context is crucial. Astronomers need to consider the type of star, the planet's size and estimated temperature, its geological activity, and the presence of other gases in its atmosphere. A single detection of oxygen, for instance, might not be enough, but oxygen in combination with methane on a planet that otherwise looks Earth-like would be far more compelling. There's also the challenge of false negatives, situations where life is present, but it doesn't produce a globally detectable atmospheric biosignature that we are currently looking for. Perhaps life on another world has a very different metabolism that doesn't produce oxygen or methane in large quantities. Or perhaps life is confined to subsurface environments with little impact on the global atmosphere. Our search is inherently biased by our understanding of Earth-based life. Beyond individual gases, some scientists propose looking for more general signs of disequilibrium or complexity in atmospheric chemistry that biological processes might create. Another intriguing idea is the red edge, a spectral feature caused by the way chlorophyll in terrestrial plants strongly reflects near-infrared light. Detecting a similar sharp rise in reflectivity at a specific wavelength from an exoplanet could indicate the presence of widespread light-harvesting pigments on its surface, though non-biological mineral surfaces could also potentially create similar features. Think of searching for biosignatures as cosmic detective work. We are looking for fingerprints left by life on a planetary scale. Each gas detected is a clue. The combination of gases, their abundances, and the overall context of the planet and its star provide further evidence. It will likely require multiple lines of evidence and probably observations from several different telescopes and techniques before the scientific community would confidently announce the detection of life on an exoplanet. The process will be one of careful, painstaking accumulation of data, rigorous modeling of all possible abiotic scenarios, and intense scientific debate. But the potential reward, the first confirmed evidence of life beyond Earth, would be one of the most profound discoveries in human history, forever changing our perspective on our place in the universe. While the search for biosignatures focuses on the chemical imprints of life, whether microbial or more complex, there's another fascinating avenue in our quest for cosmic kin: the search for technosignatures. This is the realm often associated with SETI, the search for extraterrestrial intelligence, and it involves looking for evidence of advanced alien technologies rather than just biological processes. If a civilization elsewhere in the cosmos has developed technology comparable to or significantly beyond our own, they might be unintentionally or intentionally creating signals or artifacts that we could detect across interstellar distances. What kinds of technosignatures might we look for? The classic example, and still a primary focus of many SETI efforts, is the search for artificial radio signals. Natural astrophysical phenomena, like pulsars, quasars, or star-forming regions, produce radio waves across a broad range of frequencies. However, an intelligent civilization might use radio waves for communication, radar, or other technological purposes, and these artificial signals could have characteristics that distinguish them from natural cosmic noise. For instance, a deliberate beacon or a leaked communication signal might be very narrow-band, concentrated at a specific narrow frequency, or it might contain complex modulation patterns, pulses, or information content that is clearly artificial. Early SETI pioneers like Frank Drake, who conducted Project Ozma in 1960, the first systematic attempt to detect interstellar radio signals, reasoned that certain magic frequencies, like the 21-centimeter line of neutral hydrogen, a fundamental frequency in radio astronomy, might be logical choices for interstellar communication. Modern searches scan a much wider range of frequencies, often using sophisticated algorithms to sift through vast amounts of data from radio telescopes, looking for these telltale artificial characteristics. Another promising area is the search for optical technosignatures, such as powerful pulsed laser signals. Lasers can be highly collimated, focused into a narrow beam, and monochromatic, a single wavelength of light, making them potentially very efficient for sending signals across interstellar distances, as they can outshine the host star in that narrow wavelength band, at least for brief periods. Optical SETI projects use specialized telescopes equipped with fast photometers to search for these very short bright flashes of light that couldn't be easily explained by natural phenomena. Beyond deliberate communication signals, we can also consider looking for evidence of large-scale astro-engineering projects. One famous hypothetical example is the Dyson sphere, or Dyson swarm-jock ring, first proposed by physicist Freeman Dyson. He suggested that a very advanced civilization, with immense energy needs, might construct a vast swarm of orbiting structures around its host star to capture a significant fraction, or even all, of its energy output. Such a megastructure would block the star's visible light, but would re-radiate the absorbed energy as waste heat, primarily in the infrared part of the spectrum. So, one potential technosignature could be a star that appears unusually dim in visible light, but has an anomalously large infrared excess. Searches have been conducted for such objects, though no unambiguous candidates have yet been found. Other, even more speculative, astro-engineering ideas include starlifting, manipulating a star to extract material or extend its lifespan, or planet-scale engineering that might alter a planet's albedo, reflectivity, or atmospheric composition in clearly artificial ways. We could also look for industrial pollution in the atmospheres of exoplanets. Just as human industrial activity releases certain chemicals into Earth's atmosphere, like chlorofluorocarbons, or CFCs, which are purely artificial and were damaging the ozone layer, an advanced alien civilization might inadvertently or deliberately release unusual or unexpected gases that could be detected spectroscopically. Detecting complex artificial molecules that have no known natural formation pathway would be a strong technosignature, though likely very difficult to achieve given the tiny atmospheric signals. Another category includes searching for anomalous heat signatures. A technologically advanced civilization might concentrate energy use in specific areas, leading to localized heat islands that could be detectable in the infrared. Or perhaps they utilize powerful propulsion systems for interstellar travel that leave transient high-energy signatures. The search for technosignatures is inherently more speculative than the search for biosignatures for several reasons. Firstly, while biology, at least as we know it, operates under certain chemical and physical constraints that might lead to somewhat predictable biosignatures, like oxygen from photosynthesis, the forms and manifestations of alien technology are almost entirely unknown. We are limited by our own technological imagination. What seems like a logical technology to us might be completely irrelevant or outdated to a civilization millions of years more advanced. Secondly, we have only one example of a technological civilization to study, our own, and we are still in the very early stages of our technological development on a cosmic timescale. Our own radio leakage is decreasing as we move to more efficient fiber optic and directed satellite communications. Thirdly, the question of intent is crucial. Is an alien civilization deliberately trying to signal its presence, or are we merely hoping to eavesdrop on their internal communications or detect byproducts of their activities? Deliberate beacons would be easier to find, but require the assumption that other civilizations want to be found. The Fermi paradox, which we will delve into more deeply in the next section, looms large over the search for techno-signatures. If advanced technological civilizations are common, why haven't we detected any unambiguous signals or artifacts yet? This great silence is a profound motivator for SETI, but also a source of considerable debate about the likely prevalence and longevity of such civilizations. Despite the challenges, the search for techno-signatures continues, driven by the potentially transformative nature of a discovery. Organizations like the SETI Institute and initiatives like Breakthrough Listen are employing increasingly powerful telescopes, like the Green Bank Telescope, the Parkes Observatory and the Allen Telescope Array, and sophisticated signal processing techniques to scan the skies. The approach is often to look for signals or phenomena that are non-natural or artificial by exclusion, something that cannot be readily explained by known astrophysics. Think of searching for techno-signatures as listening for a single, intelligently crafted note within a vast, chaotic, cosmic symphony. Or perhaps it's like searching for a single manufactured object, like a plastic bottle, washed up on an infinitely vast natural beach. The challenge is immense, requiring not only advanced technology and vast computational power, but also a deep understanding of all the natural noise and phenomena the universe can produce, so we can recognize something truly anomalous. If a credible techno-signature were detected, the verification process would be even more rigorous than for a biosignature. It would need to be independently confirmed by multiple observatories, ruled out as any form of terrestrial interference or unknown natural phenomenon, and likely would need to exhibit clear signs of intelligent origin, e.g. complex patterns, mathematical sequences. The societal and philosophical implications of such a discovery would be immeasurable, instantly answering the question of whether we are alone as a technological species in the universe. It would usher in a new era for humanity, filled with both incredible opportunity and profound new questions about our place and future in a seemingly populated cosmos. The quest for these faint whispers of alien technology is, in essence, a search for minds like, or perhaps vastly unlike, our own, reaching out across the silent gulfs of space. We have journeyed from the very spark of life on early Earth, through the potential habitats of our solar system, to the distant exoplanets where we seek faint biosignatures, and even further, to the ambitious quest for techno-signatures from advanced alien civilizations. Yet despite the sheer vastness of the universe, with its hundreds of billions of galaxies, each containing hundreds of billions of stars, many of which likely harbor planets, we are confronted by a profound and unsettling quiet. This brings us face to face with the Fermi paradox, a deep puzzle that challenges our assumptions about the prevalence of extraterrestrial life, particularly intelligent life. The paradox is named after the brilliant physicist Enrico Fermi, who during a lunchtime conversation with colleagues at Los Alamos National Laboratory in 1950 famously posed the question, "But where is everybody?" The context was a discussion about the high probability of Earth-like planets and the likelihood of life evolving elsewhere. Fermi's simple yet powerful question highlighted an apparent contradiction. The argument for abundance. The universe is incredibly old, around 13.8 billion years, and vast. Our Sun is a relatively young star in a galaxy that has existed for billions of years before it. There are billions of stars in our Milky Way galaxy alone, and a significant fraction of them are likely to have planets. Even if only a small percentage of those planets are habitable, and only a small percentage of those develop life, and only a small percentage of that life evolves intelligence and technology, the sheer numbers suggest that there should still be many such civilizations out there. The expectation of detectability. If even a few of these civilizations have developed interstellar travel or communication capabilities, even at sublight speeds, it would only take a fraction of the galaxy's age to explore or colonize significant portions of it. Or if they produce detectable techno-signatures, like radio waves or large-scale engineering, then we should have seen some evidence of their existence by now. Signals, probes, artifacts, or even direct contact. The observation of silence. Yet despite decades of searching, primarily through SETI, we have found no unambiguous, widely accepted evidence of extraterrestrial intelligence. The cosmos appears to be silent, at least to our current detection methods. This stark contrast between the high probability arguments and the lack of observational evidence is the essence of the Fermi paradox. It's not a formal logical paradox in the strictest sense, but rather a powerful expression of surprise and a catalyst for exploring why this great silence might be. There are numerous proposed solutions or explanations for the Fermi paradox, which can be broadly categorized. Category 1, they don't exist or are extremely rare. This category suggests that one or more of the steps required to produce a detectable technological civilization are incredibly improbable. This is where the concept of the Great Filter comes in. The Great Filter is a hypothetical barrier or evolutionary step that is so difficult to overcome that it prevents almost all life from reaching a stage where it becomes technologically advanced and detectable across interstellar space. The filter is behind us. Perhaps the origin of life itself, abiogenesis, is an extraordinarily rare event. Or maybe the transition from simple prokaryotic cells to more complex eukaryotic cells was the filter. Or the jump to multicellularity, or the evolution of human-level intelligence and tool use. If the Great Filter is in our past, it means humanity is exceptionally lucky and rare, perhaps one of the very first, or even the only, technological species in our galaxy. This would explain the silence, but it places a heavy burden of uniqueness and responsibility on us. The filter is ahead of us. This is a more ominous possibility. Perhaps many civilizations arise and develop technology, but they inevitably encounter a filter that leads to their self-destruction or collapse before they can engage in widespread interstellar activity. This could be nuclear war, catastrophic environmental damage, uncontrolled artificial intelligence, or some other unforeseen consequence of advanced technology. If this is the case, the silence we observe could be a warning about our own future. Category 2. They exist, but we haven't found them yet, or can't recognize them. This category assumes that extraterrestrial civilizations are out there, but for various reasons, we haven't detected them. We haven't looked hard enough or in the right way. Our SETI searches have only covered a tiny fraction of the cosmic haystack in terms of stars, frequencies, signal types, and time. Perhaps their signals are too weak, too intermittent, or use technologies we haven't yet conceived of, e.g. communication via neutrinos, gravitational waves, or unknown physics. They are too far away. The galaxy is vast, and even if civilizations are common, the average distance between them might be so great that communication or travel is impractical, or their signals are too attenuated by the time they reach us. They don't want to be found or don't care about us. Perhaps advanced civilizations choose to remain silent or hidden, perhaps following a zoo hypothesis, observing us without interfering, like we observe animals in a wildlife preserve, or a prime directive not to contact less developed species. Or maybe they are simply too alien, too advanced, or too preoccupied with their own concerns to bother with a relatively primitive species like ours. They might exist in forms or dimensions we cannot perceive. Their timescale is different. A civilization might only be loud and detectable for a relatively short period of its existence. If the average lifespan of a detectable technological phase is short, the chances of two such phases overlapping in time and space might be low. They are not biological or are post-biological. Perhaps advanced intelligence evolves beyond biological forms into something like artificial intelligence or disembodied consciousness, with different motivations and methods of interaction, or lack thereof, with the universe. Category 3. They were here but we missed them, or misinterpret the evidence. This category suggests that evidence of extraterrestrial visitation or activity might exist, but we either haven't recognized it, have misinterpreted it, or it occurred in our distant past. This often ventures into more speculative territory, e.g. ancient astronaut theories, but from a purely logical standpoint, it's a possibility to consider. The Fermi paradox is not just an academic puzzle. It forces us to critically examine our assumptions about life, intelligence, technology, and the future trajectory of civilizations. Each proposed solution has profound implications. If we are truly alone, it makes life on Earth incredibly precious. If civilizations inevitably self-destruct, it serves as a stark warning. If they are out there but deliberately hiding, it raises questions about their motives and our own desirability for contact. Think of the Fermi paradox like this. Imagine you arrive on a seemingly deserted tropical island that has all the perfect conditions for human habitation: fresh water, abundant food, good climate. You would naturally expect to find people, or at least signs of past inhabitants. If you find absolutely nothing, you'd be puzzled. Where is everybody? You'd ask. You'd then start hypothesizing. Was it too hard to get here? Did something wipe out previous inhabitants? Are they hiding from me? Am I looking in the wrong places? The island is the galaxy, and we are the puzzled explorers. The ongoing search for biosignatures and technosignatures is, in a way, an attempt to gather more data points to help resolve the Fermi paradox. Detecting even simple microbial life elsewhere would suggest that the first filter, abiogenesis, might not be so great. Detecting a technosignature would tell us that at least one other species has made it through many potential filters. The continued silence, however, deepens the mystery. The paradox reminds us that we are still in the very early stages of understanding our cosmic context. Our knowledge is limited, our perspective is shaped by our own unique evolutionary history, and the universe may be far stranger and more complex than we can currently imagine. The Great Silence is perhaps the most profound observation we have in the search for extraterrestrial intelligence, precisely because of what it doesn't say. It's an unanswered question echoing across the cosmos, inviting us to listen more intently, to search more broadly, and to think more deeply about our place within it all. As our deep sleep voyage through life's origins and the search for cosmic kin draws towards its final gentle drift, let's allow our minds to expand one last time, to reflect on the sheer diversity of life that could exist beyond Earth and the profound, world-altering implications of one day discovering that we are indeed not alone in this vast universe. Having confronted the great silence of the Fermi paradox, we now turn to the equally compelling what if? What if that silence is broken, not necessarily by a booming radio signal, but perhaps by the subtle confirmation of microbial life on Mars, or the spectral whisper of a biosphere on a distant exoplanet? Envisioning potential alien life requires us to stretch our imaginations beyond our terrestrial biases. While our search for biosignatures is often guided by life as we know it, carbon-based, water-dependent, the universe offers a staggering array of chemical and environmental possibilities. We've touched upon the idea of alternative biochemistries. Could life elsewhere be based on silicon instead of carbon, thriving in environments too hot or too cold for carbon chemistry as we understand it? Could solvents other than water, perhaps ammonia, methane, or even more exotic liquids, serve as the medium for life on worlds with vastly different temperatures and pressures? If so, the very forms and functions of such life would be almost unimaginably alien to us. Imagine life evolving on a planet with significantly higher gravity than Earth. Its creatures might be low-slung, incredibly strong, with thick, robust skeletons, or perhaps they might be amorphous, spreading out to distribute their weight. Conversely, on a low-gravity world, life might be tall, spindly, and delicate, perhaps even taking to the air with much greater ease than terrestrial flying creatures. Consider worlds orbiting different types of stars. A planet around a dim red dwarf, bathed in predominantly reddish light, might have plants, or their equivalents, with black or very dark pigments to absorb as much available energy as possible. Life there might have evolved senses highly attuned to the infrared spectrum. On a planet orbiting a very hot blue star, though such stars have shorter lifespans, potentially limiting the time for complex life to evolve, life might need strong protection from intense UV radiation, perhaps with reflective skins or by living underwater or underground. What about sensory experiences? We perceive the universe through a limited set of senses: sight, hearing, touch, taste, smell. Alien life forms could have evolved entirely different sensory modalities or highly enhanced versions of familiar ones. They might see in X-rays or radio waves, navigate by sensing magnetic fields with incredible precision, or communicate through complex chemical signals, pheromones, far more sophisticated than any known on Earth. Their perception of reality, their art, their understanding of the cosmos would be shaped by these different sensory inputs, creating worldviews and intelligences that could be profoundly, fundamentally different from our own. If we were to encounter intelligent alien life, the challenges of communication, as we've discussed, would be immense, perhaps even insurmountable if their cognitive frameworks are too dissimilar. But even the discovery of simple, non-intelligent microbial life elsewhere would be revolutionary. It would tell us that abiogenesis, the origin of life from non-living matter, is not a unique-to-Earth fluke, but a process that can occur on other worlds given the right conditions. This would transform biology from the study of a single instance, terrestrial life, into a truly comparative science. We could study how life adapts to different planetary environments, what chemical pathways it utilizes, and whether there are universal principles that govern biological evolution across the cosmos. It would vastly increase the probability that life in general is common throughout the universe, even if intelligent life remains elusive. The philosophical and societal implications of such a discovery would be profound. Our place in the universe. Finding any form of extraterrestrial life would fundamentally alter humanity's perception of its own significance. If life is common, we are part of a larger cosmic biology. If intelligent life is also found, we are no longer the sole possessors of high-level consciousness and technology in our observable sphere. This could foster a greater sense of humility and interconnectedness with the cosmos, or for some, perhaps a sense of unease or diminished importance. Religious and philosophical beliefs. Many religions and philosophical systems have at their core creation stories and views about humanity's special place. The discovery of alien life, particularly intelligent life, would inevitably lead to deep reflection and reinterpretation within these frameworks. It could challenge some deeply held beliefs while potentially enriching and expanding others. Scientific advancement. The study of alien biology or technology, if discovered, could lead to unimaginable scientific breakthroughs. New understandings of physics, chemistry, biology, medicine, and energy could revolutionize our own civilization. Unity or division on Earth. The knowledge that we are not alone might foster a greater sense of global unity, the overview effect on a species-wide scale, as we confront our shared identity as Earthlings in a larger cosmic community. Alternatively, it could also exacerbate existing tensions or create new ones, depending on the nature of the discovery and how it is managed. Long-term perspective. Contact with or even just the confirmed existence of other civilizations, especially if they are much older than ours, could give us a profound long-term perspective on the potential futures and pitfalls for technological societies. It might offer hope, warnings, or simply a sense of the vast timescales over which cosmic evolution operates. The search for cosmic kin is, in many ways, a search for ourselves, for a deeper understanding of what life is, how it arises, how it evolves, and what its ultimate potential might be. It's a quest driven by an innate human curiosity that has led us from gazing at the stars in wonder to building sophisticated instruments to probe their secrets. Think of the vast unread library of the universe. Each star system is a potential book, each habitable planet a potential chapter. We have, so far, only been able to read deeply from one book, our own solar system, and have only truly understood one chapter in detail, life on Earth. The prospect of opening other books, of reading other chapters, is what fuels the ongoing exploration. Whether those chapters tell stories similar to our own, or tales so alien and unexpected that they reshape our entire understanding of what's possible, the act of searching itself expands our horizons and enriches our existence. As you drift now on the very edge of sleep, let your mind hold on to this sense of boundless possibility. The universe is ancient, and its capacity for creation seems limitless. The silence we currently perceive might just be the quiet before a profound revelation, or it might be the natural state of a cosmos where life like ours is a rare and precious gem. Either way, the journey to understand our place within it is one of the grandest adventures humanity can undertake. The search for our cosmic kin in all its forms continues, a gentle, persistent question whispered out to the stars. And now, as our 2-hour deep sleep voyage through the origins of life and the search for cosmic kin comes to its gentle end, allow the echoes of these immense questions to soften and fade into the quiet rhythm of your breath. We have traversed billions of years, from the fiery formation of our own world to the icy stillness of distant moons, from the first tentative chemical reactions that sparked life to the speculative realms of alien biologies and intelligences. The universe, in its staggering scale and profound mystery, continues its silent, ceaseless dance, regardless of our understanding. The stars will continue to forge new elements, new planets will coalesce from cosmic dust, and perhaps, in countless unseen corners, life will continue to emerge, to adapt and to evolve in ways we can only begin to imagine. The questions we've explored tonight, of our origins, of our solitude or companionship in the cosmos, are ancient ones, pondered by humanity for millennia. Know that the search for answers is a patient one, a relay of curiosity passed down through generations. For now, your own journey is towards rest, towards the deep, restorative peace of slumber. Let the vastness that once held your thoughts now cradle your sleeping mind. There is a profound beauty in the unknown, a comfort in the understanding that we are part of something so much larger than ourselves. The cosmic kin, if they are out there, are part of that same grand tapestry. Allow any lingering thoughts to dissolve like starlight at dawn. Feel the gentle pull of sleep as your mind lets go and your body finds its deepest ease. The universe will keep its secrets for another day. Sweet dreams.